we start with DNA, let's start from the basics. DNA extraction, okay, there is a cell, we all know, and in nucleus there is a DNA. But we need to extract the DNA out of it and then we can process it further, right? So there are many techniques that we use for DNA extraction. Generally one which is very common in all the laboratories, it is phenylchloroform isoamyl alcohol, that is organic technique, right? It uses organic chemicals and solvents to extract DNA and purify them. Another technique is Chilex beads, in which only single-stranded DNA is being extracted. What happens is the DNA binds to the Chilex bead, and when the temperature is increased, the DNA gets differentiated from the Chilex, and we extract that out. In Chilex technique, generally we find single-stranded DNA only, right? No double-stranded DNA has the characteristics of being double-stranded, right? So, but in Chilex technique, we will always identify single-stranded DNA. The another technique is silica column-based extraction, where the cells are there, we lyse the cells, we put the cell lysate into the column, we spin it down, the DNA will be caught by the silica column, rest all the debris will be washed away. Then we add elution buffer, right? And after adding elution buffer, the DNA gets separated from the silica column and comes into our desired tube, right? Another technique is magnetic bead extraction. There are beads which extract, there are beads having magnetic properties, and they extract DNA from the cell during lysis and make them attach to themselves. With the magnetic properties, we remove the beads and we isolate DNA from the magnetic bead, right? So these are the basic techniques that we follow in this laboratory for DNA extraction. Once the DNA has been extracted, right? So we need to know how much DNA is there, right? Or DNA is there or not, first of all, right? It's like hawa mein tuka maro, magar we need to solidify that thing with an evidence. So the technique that we use is gel electrophoresis, right? This is assembly of gel electrophoresis. It's a large unit. Just behind you is a medium unit, and there is a small unit. We select the units depending upon the sample size. This goes above, if you have samples above 100 to be tested. So this unit is being used around 50 to 20 samples. The medium is used, and within 10, the small one is used. Generally, see, this is the gel I ran today morning itself. Yeah, this is agarose gel. Yeah. A well purified agar. It's agarose. And the thing is, if you are checking the DNA which is extracted from the cell, we use generally 1% of agarose gel. Whereas a PCR reaction is done and a particular amplicon has been generated of a DNA, which is very short compared to template DNA is in kilo base pairs, right? But this PCR product may be of 1000 base pairs, 800 base pairs. It will run quickly in 1%. So we make more concentrated agar gel. That is 2%, right? As you see in this agar gel, you can see two colors. One is light blue and one is something violet, dark blue. Generally, when a template DNA has been identified, okay, the tracking dye that is being used is bromophenol blue. This is the blue one, right? It tracks the DNA, template DNA, okay. As we cannot see the DNA, we need something to visualize it so that the DNA doesn't run out of the gel. So bromophenol blue is just taking the tracking of the DNA, right? But as we know, PCR is quite smaller fragments. So we add one more dye, that is valine which runs quickly than bromophenol blue. So our identification, our region of PCR product to be there is almost over here between the bromophenol blue and the silane. Okay. This is the gel, but this gel has to be casted in this particular assembly. This is the gel casting assembly. Yeah, to like this DNA you cannot be visualized just by keeping the gel and you can visualize the DNA. We need ethidium bromide or cyber green. There are two chemicals. That is, ethidium bromide is generally quite carcinogenic, and that's why I'm wearing gloves. I'm touching the gel, but you guys should not be touching it. It gets absorbed to the skin and it is highly carcinogenic. So even with the casting plates, so avoid touching it. Don't get afraid of it. It's not that much contagious. But yes, this is ethidium bromide. What ethidium bromide does, as of the DNA is running and separating on the basis of size. Yeah, we cannot visualize it. But whenever ethidium bromide is added, there are DNAs of generally of a double strand, right? So ethidium bromide comes between, it intercalates between the two strands of DNA. So whenever DNA is running through the gel, the ethidium bromide gets intercalated between the gel, right, DNA. And when a UV light is being put on it, it gets fluorescence, right? And hence we can see our DNA. As of now, we cannot see the DNA, right? So the extracted DNA as well as the PCR is, as we run through over here, it is being observed because of ethidium bromide or cyber green. Both of them are having the same property of intercalating, right? Let's see how the gel is being casted. Okay. Yeah, this is a larger cast that is being used for the large unit, right? We prepare the banks of it. We boil the agarose in the microwave. We cool it down to the room, not to the room temperature, but around 50 degrees or 45 degrees till we don't feel the heat on our palm. Once it is comfortable on the palm, you add ethidium bromide into the agarose. We stir it gently so that no air bubbles is forming, and we pour it down into this. Once the gel has been poured, we need to make some wells for it for placing our samples. So these are the combs that are being used. Various types of combs are available. This is a 40 sample comb. This is a 28, 20 samples, 16 sample, and 10 samples are also available. The size of the comb increases, just nothing more in that. We place the comb after pouring the gel and we let it cool down. Right? Once the gel has been cooled, we remove the combs, and the gaps that has been formed, we call it wells, where we put the samples, right? And after the gel has been casted, the combs are removed, the banks are removed, we place the gel over there into the buffer. TA buffer it is used. So we put it into the buffer. Okay. Generally the sides where the DNA is being to be loaded is the negative side. As the DNA is having negative charge, it will always run towards the positive charge. So we place the sides where we want to load the DNA towards the negative charge. Okay. If we put the plates opposite, DNA will get out of the gel. That happens with us also if we do it by mistake. Yeah. So we have to make sure it's on the negative side, and we give the electric current, and as the electrophoresis principle, it gets separated on the basis of molecular. Yeah. So this is how this gel was casted and ran into this assembly, right? It was done today morning itself. So once it is done, we need to visualize it. We just cannot see the gel. We cannot see the DNA from here, right? So we use gel documentation system, right? But before going to that area, there is one instrument behind you. That is nanodrop, generally used for quantification of DNA, right? Any DNA extraction that has been done, we need to know how much DNA we got, right? So with the help of UV absorbance, at 260 nanometers, it absorbs the UV light, right? And depending upon the peak that we get, how much absorbance is there, we find how much quantity of DNA is there, right? But generally that is being used, like Lambert-Beard law is there, but it is used for 3 ml, 1 ml, right? For UV spectrometry. Over here in this instrument, major benefit is of the sample size. As hardly we get 50 microliters of DNA extract, this instrument just uses 2 microliters, right? So quantification can be done, and then visualization of the DNA. How? Let's go to the gel documentation system. Please. This is the stage of the gel documentation system. Okay. White light, there are two sources of light, white and UV light, right? White light is just to adjust the gel. The live camera starts over here. We adjust the gel accordingly with the help of the white light into the screen, and we focus the gel properly, right? And this stage, below this stage, there is UV light or UV tubes, right? Once the gel is placed upon it, we put it inside the chamber, we close it, we switch off the white light, and we switch on the UV light, right? After switching on the UV light, what we see into the gel is like this, right? So this is how we visualize the DNA. As we can see, this is a quite bright band over here. this shows that there was a DNA at that position where ETBR got intercalated between it. Okay, and because of that ETBR, we are able to visualize the DNA under UV light. Right? So this is a PCR product that I ran in the today in the morning. Okay, and this you see over here is a ladder, size standards. Okay, this ladder consists of 100 base pair of DNA, 200 base pair of DNA, 300 base pair of DNA, 400 base pairs of DNA. So as electrophoresis separates the DNA on the basis of its molecular size. There are standard ladders which it will separate. When a particular DNA comes at a particular coincides with a particular ladder, okay, for example, it coincides with 700 base pair of DNA, right? So we can approximate our base pair of DNA. Generally in PCR, we know the amplicon size, whatever it is going to generate. So if it doesn't come on the expected amplicon size, for example, I am expecting that my primer is generating. I hope PCR is clear.
Yes.
Basics of PCR, primers are there, forward, reverse, right? Yeah. So in a PCR, if my product is getting generated of 500 base pairs, I'm according to my calculation, it should be 500 base pairs. But over here it comes 700 base pairs. So there is a chance that it is getting non-specifically binded somewhere else. Right? Either that particular DNA is not getting amplified or there is non-specific binding of the primer. So the DNA ladder helps us to determine the approximate base pairs of our DNA. Right? So this is how the gel is being viewed into the gel documentation system, the DNA, how it is visualized. Any questions regarding, please feel free to ask. Any questions?
This is the result on which base of which you will give your report, DNA fingerprint.
No. That comes in lab 3. That will go. Okay. So any questions till now? Please feel free to ask.
It is just like a X-ray.
It is just a raw visualization technique or method that we use just to see whether our DNA is coming or not.
No, there is no.
Right? First we need to see कि भगवान आया कि नहीं, and भगवान आया तो कितनी क्वांटिटी में भगवान आया, क्योंकि वैसे भी वो दिखने वाला है नहीं, as it is very in small quantity. Then we can start playing with our DNA.
Yes.
Right? So that is being done in lab.
This is the step where we found that we got DNA or not.
भगवान प्रसन्न हुआ. It is the step where we find that it is there. So we head towards lab 3, right, where more advanced instruments are there.
Thanks.